introduce our next speaker. Maggie is a second-year student in the MCSP program. She is in Marcus Seldin's lab, and she is going to give us a talk on ICA. All right, Maggie, take it away.
All right, I'll share my screen. Oh wait, it's disabled. Someone help me out with that so I can share my screen.
Should work now.
All right, here we go. Okay. And okay, all right, let's get started. Can everyone hear me okay? I assume that's a yes. Okay, so my name is Maggie Myers. I'm in Marcus Seldin's lab and John Lonegrubb's lab. Marcus is a pretty new faculty member that studies systems genetics. So today I will be talking about ICA decomposition as a module detection method. ICA stands for independent component analysis. And as a general outline for this presentation, first I'll talk about how ICA came up on my radar, how it works, applications to transcriptomic data, and then some examples with post-processing. If you have any questions, just feel free to jump in at any point. So module detection methods are useful when you have a large data set of the full transcriptome and you want to figure out what's happening at a larger systems level. So basically grouping genes into modules of co-expressed genes that typically represent pathways. Usually people tend to use clustering methods for module detection. The most common example is WGCNA, but there are a few drawbacks of clustering methods. So first, genes can't be assigned to multiple modules. And we know that this is a problem because it's been shown many times that genes participate in multiple pathways. And so we know that there's overlap between modules. Second drawback of clustering methods is that these tend to look at global patterns of co-expression, but in reality, gene expression tends to be context dependent. Samples will show some patterns of co-expression in genes, and when we use clustering, it can't pick up those subsets of samples. And third is that these clustering methods tend to ignore the regulatory relationships between genes, like transcriptional regulation. So the first two drawbacks, these are basically solved by methods such as ICA decomposition and biclustering. And then you have network inference methods that tries to solve that third problem of clustering. So Marcus sent me this paper, which I'll just briefly review, that convinced me to try ICA decomposition because we were searching for ways to basically overcome these drawbacks that we get when we use clustering methods for module detection. So this paper, love this paper, highly, highly recommend it. I won't go too in depth just because, you know, it's not the main point of this presentation, but I do highly encourage you to read it. So first I want to go over their methodology that these authors used to evaluate module detection methods. They tried 42 different module detection methods. Most of these were different methods of clustering. There was biclustering, direct network inference, decomposition methods such as ICA, and iterative network inference. And they used 9 different expression data sets from 3 different organisms and also a synthetic data set that they used. And they came up with a new evaluation metric to compare observed versus known modules because existing metrics can't handle overlapping genes. And so I thought this paper was pretty thorough and comprehensive in their approach. And so they did a pretty good job convincing me that what they were saying was true. These are the results. And so here, this subfigure A is, so a higher score basically means that it's better at detecting known modules. And as you can see, so we have WGCNA, which is pretty common. It does a pretty good job. And so clustering has a pretty high score. You can see the ICA decomposition, which is right here, really has the best score and is the best at finding known modules. In addition, this can also handle overlap. So genes can be in multiple modules, and it can look at local co-expression instead of only global patterns of co-expression. And also, if you want to look down here at the runtime, which I know, once we get into analyzing big data sets, this does become important. ICA runs pretty dang quick. Actually, when I ran it, it takes less than 5 minutes. It's really amazing. So, yeah, so these are some of the results that really convinced me to try out ICA decomposition. So, what is ICA? So this is independent component analysis. So suppose that you have, so this can be, for example, if you're at a cocktail party and there are multiple people talking. And so you record these conversations and each signal can be a voice. This is just a simple example. And so mutually independent as possible. So everything hinges on the assumption that these components or modules or signals are statistically independent. And so this method tries to describe how the recorded data are generated by the mixing of individual components or signals. Are there any questions so far?
How is this different from Fourier analysis?
I'm really not sure. I don't know too much about Fourier analysis.
It just sounds like a similar thing where you're like, you're decomposing into an orthogonal basis, essentially.
Yeah.
So I was just curious.
Do they have to be orthogonal in Fourier analysis?
Yes, by construction, they're exactly orthogonal.
I think that might be the difference, because Maggie, correct me if I'm wrong, but my understanding is these don't need to be orthogonal. Is that
Yeah,
correct?
they're non-orthogonal. I think that's correct.
Right.
Yeah, so ICA can be considered as a rotation of PCA axis, so principal component analysis, after data whitening, which is orienting the Gaussian ellipsoid along the coordinate axes and scaling them to unit variance. And so I know that Emmanuel told me he was going to ask me about the differences between PCA and ICA, so I'm going to go ahead and get that out of the way. So PCA, principal component analysis, the overall goal of PCA is to compress information, whereas for ICA, the overall goal is to separate information. And PCA, it finds projections to minimize the reconstruction error. So the variance of the projected data wants to be as large as possible. And PCA only uses up to second order moments, so the covariance, to use this method. ICA, on the other hand, finds directions in the data that maximize independence. And they want the projected data to look non-Gaussian, so they're as independent as possible. And this is more likely to align with the data. And in order to get this data as independent as possible, ICA uses higher order moments, not just second order moments. And in PCA, some of the components are more important than others. These are based on the eigenvalues, whereas in ICA, all the components are equally important. So applying ICA to transcriptomics data sets, it hasn't been done very often. I've noticed in my research of ICA that it's often applied to like magnetic resonance and other things, but not as frequently to transcriptomics. And so this paper up here was pretty useful in trying to understand ICA in the context of omics data sets. So you have your data, which is the columns are genes and the rows are samples. And you run ICA and you get two matrices. One is your samples and components. And these columns of this matrix, these are metasamples. Then you have this is your matrix S, and each row is a metagene. So each metagene, which is kind of congruent to a module like in WGCNA, it's also called a component. These metagenes represent a molecular program by assigning a weight to each gene. So the weight of each gene in a metagene will tell you how much that gene contributes to the metagene. And you can think of these metagenes as a molecular program. And metasamples, so the activity of metagenes and sample is combined additively, and each metagene acts on a sample with a sample-specific strength or activity. So activities of the same metagene over all measured samples is a metasample, and a metasample is the profile of the corresponding metagene activity. Are there any questions so far? All right, cool. All right, so to use ICA, the best way is using this Python package. It's called Fast ICA. There is a package in R, but it's not as good. And so this chunk of code up here is, so besides the post-processing, this is all it is. And this runs within 5 minutes, which is really amazing. So data, and then you have these 3 lines of code to do all the ICA. And then you get your matrix of the metasamples and your matrix of the metagenes. And so, which I forgot to include this later in the presentation, but one way that you can do post-processing is you can look at the distribution of each metasample. And so that can point out if there is a molecular program that's expressed in some samples, so in some individuals, but not in others. And so for the data set that we've been using the GTEC data set, which has bulk RNA-seq from many different tissues. So there are no instances of like a bimodal distribution in metasamples, but if there was, that would be cool. And so for finding the number of optimal metagenes, so this is something that I haven't quite gotten to yet, but I've seen recommendations of using the false discovery rate estimation in R to detect potentially overlapping modules within each metagene. So when you run ICA, you have to give it how many components that you want it to estimate. And for post-processing tools, specifically looking at ICA for transcriptomic data. So just as a disclaimer, I am kind of bad at finding useful things on the internet. So there could be some tools out there that I just haven't found, but there's like weirdly like not that many tools out there. So the only thing that I did find for post-processing tools was this thing called Biotica, but it hasn't been updated in 5 years and it's mostly in Java. And so I figured it might be more trouble than it's worth. And so what did I do? I wrote some of the code myself. And so it's still something that I'm working on, but I do have a good bit done. I don't have it on GitHub yet, but if you contact me, I can send you my code and I'll have it on GitHub like ASAP. So how do we analyze these metagenes? So this right here, this is the distribution of the weights in a metagene. And so looking at, I sort of looked at what the package Biotica, like what they did to do their post-processing on ICA results. And they recommended, so the mean plus minus 3 standard deviation. So outside of this interval is statistical outliers. And so these outlier weights belong to genes that significantly contribute to the metagene, or they drive this metagene. And so if this metagene is a pathway, these would be basically the genes that drive this pathway. And so note that this y-axis, this is a log scale. And doing this, getting the genes outside of this interval, filters out over 99% of genes. In the code that I've written, you can change this number to get either a larger set of genes or a smaller set of genes, depending on how much you want to see. So note that the genes have positive and negative weights. Most of these weights are positive, and so the signs of the metagene can be flipped without affecting the model. That's something that I learned like 2 days ago. And but Biotica recommended to look at these positively and negatively weighted genes separately. And so, and I don't have these results to show you here yet, but something that we did when we were analyzing our GTech data with ICA and post-processing is, so in our GTech data, we have genes from different organs in the body. And so I plotted the composition of like the organ composition of genes that are positively weighted and genes that are negatively weighted. And these tend to be very different. And so there does seem to be almost qualitative differences in the genes that are positively and negatively weighted. And it's something that I want to look into more. But with this method, you should be able to flip the signs of the metagene without affecting the model.
So in terms of looking at like the metagenes in terms of the positively and negatively weighted genes separately, is that just like analogous to basically like splitting up a module?
In a way, yes. So the paper that I talked about at the very beginning, as grouping them all together. And so these genes do seem to be like in the same module, but I do want to go in and sort of, really get a list of all these genes and see if there are known relationships between the positive and the negative genes to just confirm whether they are, in the same pathway.
So just to be super clear, are you saying that, so do you know what the interpretation is if you have positive or negative weights?
See, my initial interpretation was that so positively weighted genes would be genes that are more upregulated in some samples, and then these would be kind of like offset or balanced by the negatively weighted genes. So those genes would be have a lower expression in those samples. Does that make sense?
It does, but did you confirm that?
I did not yet confirm that.
Okay.
Yeah, that was my initial interpretation. But then I saw like 2 days ago that you can flip the sign without affecting the model. But yeah, but that'd be something to explore.
Yeah, I would even, I mean, I suspect you might be able to get farther if you, I don't know, I guess this is just what I would think, but consider taking a mathematical approach versus a biological approach, like go into the ICA literature and be like, okay, if things are positive, what does that imply? Do you know what I'm saying? And then try to backtrack to the bio.
Yeah.
But this is great. Cool, thanks.
Cool. All right, any more questions before we move on? All right. Okay, so looking now at overlapping genes. So as we move forward, we're basically going to assume that we have filtered out these genes within this interval. So now we're kind of only looking at genes that are outliers in each metagene. So we're just looking at the top contributing genes now. So now So I, in particular, wanted to investigate overlap between these modules because that is one plus of ICA is that genes can be in multiple modules. And so this is a histogram of basically, so it's the number of metagenes that that a gene is part of. So as you can see, most genes are only contributing to one metagene, but there are some genes that contribute to. And for our example, I used 64 components, so 64 metagenes, and that number is just because we did WGCNA a couple of months ago and it had 64 modules. And so we just used 64 metagenes for comparisons. And so you can see that there are a small number of genes that are part of many different metagenes. So to investigate the genes that bridge or overlap metagenes, some of my code creates a data frame of metagenes with only the top contributing genes, and it's ranked by the degree of overlap. And so for some preliminary results, so as I said, we've been using this GTEC data set. So it's bulk RNA and multiple tissues. So in these genes, you have your gene name followed by your organ name. And you can probably just ignore these very last things. These are like the specific tissues, but we've mostly been paying attention to the organs in the genes. And so these genes are ranked by their degree of overlap. So this first gene is one of the top genes in like all 64 metagenes. And so the goal of this project was to try to look at communication between tissues. And so we were hoping to see some endocrine regulators, and we did. We have a GH1 from the pituitary, and some of these I don't know much about, but Marcus has told me that this looks like, we couldn't have like made up better data. It's pretty interesting how many endocrine regulators are overlapping the metagenes.
So I have a question here just about data organization, which is I'm not sure how you ended up with gene underscore like sample that it originated from. Can you explain how you got from like your gene expression matrix to this?
Yeah, so it was something which I believe that Marcus did most of the like prepping the data. But so there's these genes, and the expression of all of these genes are measured in multiple different tissues. And so this right here, this is the expression of CPA2, but only in the pancreas. And so the actual data set is like very, very huge. But yeah, does that kind of answer your question?
I'm just a little confused because I guess I would have imagined that like a gene that belongs to multiple modules is probably expressed in multiple samples. So I'm a little confused on like the specificity of like the sample to gene mappings.
Yeah, so I mean, each sample like is like one like individual person. And so you have like CPA2 in the pancreas and CPA2 in other organs, like all within the same sample. So the samples are people, not organs.
Right. Okay. Don't worry about it. I'll see if I can ask you later because I'm just a little confused by this. I would just basically my understanding is that from what it sounds like you'd just get a list of genes, not like a list of gene sample or organ, like for computing the genes with the highest overlap. Yeah. Anyway, I'll bother you later though.
Okay. All right.
I have a similar question. Like, so what you use gene weights? Like, what is this weight exactly? Like, is the weight this gene embedding thing? Is the weight, or what is the weight? Like
the weight is. So can you repeat your question again?
So what exactly mathematically weight means in this context?
So
how they calculate the weight?
Okay, so the weight, so we don't really talk about the weight much on this slide, but the weight of the metagene So for the gene that has the highest weight in the metagene, we can assume that that gene is a driver of that metagene. So if a metagene is a pathway, then the genes with the highest weight, we can assume that they are genes that most control the pathway.
So like we have this number of dimensions. For
Mhm.
each dimension, we call each dimension a metagene, and we only consider subset of genes affected in this dimension, and then we get the most affecting gene, like most important gene,
Mhm.
based on their contribution score.
Yeah. And so, so this that I have here, so this doesn't consider like if a gene has the highest weight or not. These here just consider if a gene is has a weight that is a statistical outlier. So it means it's like a relatively extreme value.
Statistical outlier compared to other samples, like this weight
Yeah,
of the
compared
samples
to other
are.
weights in the gene. Yeah, so if this is the distribution of weights in a metagene.
But when you compare to other genes, not other samples,
Compared to other samples?
like do you use some? Okay, so like this gene have higher weight compared to other genes in the same metagene. Not this gene don't have a higher weight compared to weights of same gene on another sample.
By sample, do you mean like organ, or do you mean from an individual?
The individual. So we don't compare individuals, we are just comparing genes in the same metagene group.
Yeah. So we're not comparing like individuals and samples. So if we wanted to bring that into our analysis, then we would want to look at these metasamples. So basically, like an index. So like, let's say we want to look at metagene 2. We would look at this second element, the second like metasample, and that would tell us that in that individual, this is the overall sort of expression of the second metagene. And so the metasamples tell us how much a metagene is expressed in an individual.
But we are doing it for each sample then, or because like this matrix is like, okay, so in here we have like a gene, metagene, and n number of samples. So we are focusing to each value in each sample, or?
So we're not really focusing on samples because these metagenes, I mean, this represents a molecular pathway, and this molecular pathway, I mean, if we want to do a sample specific thing, then we can incorporate these values from the metasample. But in terms of just looking at what's happening in a metagene or a molecular pathway or a functional subsystem, we only look at the weights in the metagene. Does that answer your question?
I think if I understand correctly, she analyzed all of the samples together.
Mm-hmm.
Like this is an aggregate.
You are using the second matrix, the S matrix. You are using the S matrix.
Yeah, I'm using the S matrix.
And then you're going over your number of latent dimension, so number of components. You have like let's say 64 components. You call this metagene.
Mm-hmm.
And then you analyze.
And then you analyze the weights, the distribution of weights in each metagene, and you see what genes have the most extreme weights in a metagene.
So you're doing it on a per metagene basis.
Yeah.
Okay, got it.
Yeah, so you iterate through all of the metagenes, and basically you look at which weights have statistical outliers, and then you figure out what genes have those outlier weights.
So metagene is a vector of size.
Of size, however many genes you have.
Okay, so okay, I see now. Metagene is the vector of, and then those are the weights. Then you get the top weights for each metagene.
Yeah.
Okay.
Okay.
I think we should move on.
Okay.
Okay.
Yeah.
All right, let's proceed. Okay. So something else I did was figure out how to plot this network. And so these are the 64 modules, and I use the package network X in Python. And so when I was thinking about, what should the edge weights indicate, so for this network, I only considered how many overlapping genes there are between two metagenes. So for each metagene, you have a set of genes that have extreme or outlier weights. And then you see how many genes are in common in that set between two metagenes. And then I had to kind of put a threshold on there because there would be a line between everything. And so I think I plotted, this is like the upper 40%. And so these are in the edge weight corresponds to the number of overlapping top genes. And so I just made this plot a few days ago. I want to kind of dive more into this and really figure out what's happening. But yeah, this is some code that I wrote, and I really wish that I had this on GitHub so I could, have everyone go look at it if they want to. But it'll be up there soon, or you can reach out to me.
What's the blue and the red edges?
Oh yeah, so the blue and the red edges. So this was before I found out that if you flip all the signs of the weights in the metagene, it's basically the same thing. And so the blue lines, those correspond to, so for example, if the shared genes here are both like have a positive weight and this has a positive weight, so they're the same sign, so it's blue. But if one has a positive weight and one has a negative weight, then they're red. And so basically that looked at, is it same sign or opposite sign for the shared top genes. Yeah, so you can kind of ignore the colors because I'm still trying to figure out, what the positive and negative weights can mean, or if they do mean anything.
Maggie, can I ask you a quick question? So
Yeah.
you're spending a lot of time talking about the meta genes, right? But
Yes.
you're also, are there interpretations of the meta sample that
So
would be interesting to analyze?
Yeah, so the meta sample, so for example, if you have a data set where you have like some non-diseased individuals and some diseased individuals, and if you analyze this, then you might see some meta samples that have a bimodal distribution. So basically, if some samples express a metagene more than others, then that might indicate a pathway that is associated with a disease.
So, and I guess in the parlance, it's kind of like soft clustering.
I guess so. I'm not sure,
yeah,
but
yeah, I think so. Okay, no, that's cool.
It was in the
very cool.
figure Maggie showed slides earlier. The paper compares ICA to soft clustering. So yeah,
Okay.
they're the same.
okay.
Excellent. Thank you, Maggie.
Yeah.
Like, do you correct for confounding factors?
No, I do not.
I mean, I think the outliers you get maybe genes which are differentially expressed in like male or female or different populations, or like I don't know, maybe likely to be like that.
Yeah,
And
because then that would probably come up in the distribution of a meta sample. If it was expressed differently in like males and females.
So Hassan, one of the reasons that people like to perform these sort of like module algorithms is literally to like find which modules correspond to like these differences. So even if you're like comparing disease versus non-disease and you have males and females in your data set, it is informative to know what the differences are between males and females within your samples too. So those aren't things that you, I mean, as far as I know, and I've only run WGCNA like once, those are things that are informative, even if they're not your goal from doing these sort of module finding algorithms.
Mm-hmm.
I believe people correct for those confounding factors before they do any statistical analysis.
You can basically regress out any confounding factors that you wish to at the gene expression level, like just using, I don't know, like linear or logistic regression in R. Usually we like, at least in my data in the brain, we care about like post-mortem intervals, like how long has the person been dead before we froze their brain. And that's usually something to account for. Other, I don't know, age, sex, usually we account for that before we do module detection.
Any other questions? Okay. All right, so for future work, so I want to incorporate some pathway enrichment or gene ontology for the metagenes to see what pathways are represented. And then also use that in conjunction with the network plot to see if we can elucidate anything. I also want to find some other ways to plot the network. So for example, the network that you saw on the previous slide, that only considered the number of overlapping genes, but it didn't consider the gene weights. And so as we know, some genes can have a super duper high weight and be like the driver of a metagene, and some can have relatively lower weights, but still be a statistical outlier for the metagene. And then I also want to use the false discovery rate estimation to optimize the number of modules and also make my code a little bit neater and more accessible. It would be nice to get this on GitHub and actually, create a resource that people can use, because there's definitely a gap. It was, pretty difficult to find anything for, analyzing transcriptomic stuff with ICA. And just want to say thank you to GenPALS for inviting me to speak. Really appreciate it. And feel free to reach out to me for any code, or if you have any questions. Here's my email, and code will be on my GitHub soon. And I also want to thank my two PIs, Marcus Seldin and John Lowengrub, for their continued support. All right, any questions?
When you post the code on GitHub, will you notify everyone in the Slack?
Yes, I will.
Yay, thanks.
Awesome.
Maggie, I have a dumb question.
Yes.
At the start, you said that there's ICA in R, but it's bad. So why is it bad?
I haven't tried it in R because I kind of hate R. But I know that
totally fair.
I've read that it's a good deal slower than ICA in Python. I mean, it probably still works as well, maybe, but yeah.
The other thing I wanted to ask is like, do you want to go back to that slide at the start with like the comparison of the scores? Yeah, wait, you passed it from that like review paper or the benchmark paper. So like, okay, I think that ICA is clearly the top, but like these scores seem like it's hard to get a sense for like what this means. Like, how is 8 so much better than like 7? Like, doesn't it look like like most of the clustering, like I think like in their conclusion in this paper, because I like have read this paper, and I think that they say like, oh, you can do like a few of these are pretty good, but like they're not just gonna say like ICA or nothing, right?
Yeah. Yeah, I mean, WGCNA is still, pretty good, and there's other methods that are still pretty good, but, this was at least a paper that really got me wanting to try ICA, because something that we're really interested in is looking at the overlap between modules, and you just can't really do that with WGCNA and other clustering methods. And so in terms of module detection methods that can handle overlap, ICA seems like a very good option.
Have you found that like the genes in the different like, do you think you're like over clustering with 64, or do you think 64 is like a reasonable number?
Yeah, that's a good question. That's something that I have been wanting to look into, because I still have yet to get the FGR code going to see if any metagenes can be broken down into smaller modules. But it's possible that 64 is too much or too little.
I feel like with your data set, like, because you're using GTEx, which has so many different tissues, that like there probably are a lot of modules, but if you're just looking at like one tissue, then it might be a lot less. So like, yeah.
Like when you do this, you merge all the tissues into a single metric, like metrics,
so each tissue has, so you're going to have data for gene expression for every gene in every tissue. And so you have, it's a lot more data than
your input is number of genes time number of samples and number of tissues, like genes versus samples time tissues, or like genes versus tissue or genes versus sample.
It's I might have do I have, so just for example, so like these rows are different patients, so different individuals, and so you have this gene in adipose in this tissue, and then that same gene in the adipose but in a different tissue, and then basically the same gene in multiple tissues, and you have those measurements for multiple different people. Does it answer your question?
Yeah.
Okay, great. Other questions?
I have a really stupid one.
Go ahead.
How do you know that the overlaps you detect are like true overlaps and not like an artifact of the algorithm?
I would like to think that just based on some of our preliminary results. So in particular, we're looking at, we want to study how endocrine factors might be responsible for communication between tissues. And just based on seeing, some of these endocrine regulators as most like the most overlapping genes, it seems like it's doing, kind of the right thing, in that this overlap, seems biologically reasonable.
Right. And so if you run the other method, do you just not see these results, or is it you can't get these results basically, right? So
are you talking about the overlap?
Yeah, so when you run, if you ran WGC or whatever the hell it's called on this data, and you wanted to try and make like similar conclusions, I'm trying to think of how you would benchmark this method essentially. Like,
so so in WGCNA, each gene is assigned to exactly one module. So they put it in one pathway. I mean, you can see like which genes have like the most connectivity within a module, and you can also see like correlations between modules, but you can't really see like you know, which genes connect two modules or more than two modules.
So I guess my question is like, so my question now is like, if you didn't know ICA existed and you wanted to ask the same question you currently, and you only knew WGCNA existed, what would you do? Like, could you even answer that question?
Which question? Like the question of overlap?
Yeah, overlap or looking at endocrine regulators. Because you're right, you could take two like distinct classes and then do like correlation or cosine distance or something, right? So like,
yeah, I mean, you probably want to look at like interconnectedness in a module, and yeah, that is a good question, because that, you know, one of the reasons that we started doing ICA was because it was difficult to answer that question for WGCNA.
okay.
And I haven't done a ton of WGCNA, and so, you know, if I was an expert at it, I might be able to answer that question. But
yeah, I was just getting the impression from what everyone was saying in this talk that anything you do will be compared against that method. So I was just curious.
Yeah.
So I actually think that's an interesting question, right? So I guess did you so WGCNA also gives you modules. It's just that it groups things into like you can only have one gene per module, right?
Or like one gene, like every gene goes in exactly one module.
Right, exactly. So I guess just to follow up on Axel was saying, did you look at, I guess an assumption that you can make, which is maybe a bad assumption, but assumption that you can make is that the gene that has the highest weight in a module in ICA would be in that module for WGCNA. Would that be a good assumption to make? And if
Yeah, I think so.
Okay, and that's kind of testable, right? Like you don't assume that the modules are exactly the same, right? But couldn't you look at the correlation of that way? I don't know how WGCNA works.
Yeah, I think that's something that you could test, which is something to put on my to-do list.
That's cool. It's really neat.
Like, can we draw in this presentation or kick? Like, I have a question, but maybe I should explain it visually. Like, can I draw or?
Yeah, I can. If you want me to, I can like stop sharing my screen and you can share your like whiteboard screen.
Yeah, I can try it. Okay. Sure. So do you see my screen? Okay, so like that is your number of samples, like S, and those are number of genes, and you multiply these and you get your like you basically do factorization, and that's your original sample versus gene matrix. So basically your like this method like stores specific information in each column. Like we have samples represented as a number of n dimensions. So each component in a sample should store a specific information. So let's assume that it is not always clear, but let's assume that like this column contains information about sex. And then basically then you have each gene here. And let's say get this gene I. So this gene I, so given a gene I, we try to predict, so we have the same gene I over here. So given this target vector, we're trying to fit a linear regression for gene I against each sample. So based on this linear regression, we get this target value. So those are the coefficients of linear regression. So here is the.
So what does the data look like in S? Is this like what?
S is it's your samples, like sample your sample factorization.
Meta samples, right?
Meta
Oh, okay,
samples
your
and
meta
meta genes.
samples.
So
Okay.
basically what's going on here, for a given gene I, you have this beta, and it's your X. So X time beta, you try to find yi. Yi is the vector of this thing, yi.
Yeah, but
So-
see, if S is your meta samples, I mean, I don't think you're going to have a meta sample doesn't correspond to like a particular trait like sex.
Yeah, but
I mean,
yeah,
you can-
that's correct. But there is a columns of this meta sample should denote specific features which shared cross samples. So if this sample A S1, there's 100 components in here should describe this sample. So, and since let's say age or sex is very important genetic signature, one of these component or few of this component should describe this thing, and hopefully one of these components should, or few of this component, give us information about disease, or maybe if this from multiple tissues, maybe it's just specificity, but like this information.
Yes, no, essentially this is correct, Hassan.
So yeah, so basically what you can do is you can get this metric and you can correlate this against, so you can build another regression model and you can check what kind of information stored there. So you can get these metrics and you can fit a, let's say, logistic regression to predict sex, or you can fit a linear regression to predict age. So you can actually see how much variance for different traits this sample metric explains. So then we can know exactly what information stored in here. And like I have few prior experience, and I can tell you if you have 60 components, first 30 of them will be confounding factors. The most strong components are usually confounding factors, and you can basically see that by fitting a linear regression using this metric, rather than like fitting. You get this metric X and try to predict different confounders, and you see that this like certain components, even you can get a future importance. You can check the weights of this linear regression, and you see that certain components strongly relate to specific traits. Then, since let's say the second component relates to specific trait, you can know that from the weight of this linear regression. Then what your meta component, because since second column in here in the matrix multiplication like corresponds to second row here, because you multiply this here with this here. So then this meta component also will be meta component of sex. And then
So Hassan, all of this is accurate. My question is, are you going to ask Maggie a question?
yeah, so my question is, I think you should do it's more criticism than question. So I am very worried that you are learning confounding factors for samples, and then your meta genes are the set of genes controlling these confounding factors, and the top genes you find can be the genes explaining these confounding factors. So I'm quite worried about this approach. I just want to share that.
I mean, I feel like that's, again, going to be a problem in any software that's trying to find these modules, right? Like it's going to learn the confounding factors if you don't remove them. But of course, if you do remove them, then it won't.
Plus it also, so I don't know, I think machine learning people have a specific definition for what a confounding factor is, but my point is maybe we are interested in the differences between the sexes. Do you know what I'm saying? So really there is that, that's a huge problem in science, obviously, but like what and I think a broader point is you are correct in that these meta samples identify, column, maybe groups, right? But they could be combinations groups, right? So you could have one column that's like sex and disease and a bunch of other stuff, right? So I think it's a point. I think it's a valid point. It's probably something that Maggie can look at. I don't think it's a criticism, simply because if it's something that we're aware of, then we can do essentially what you described and look at the correlation of the meta samples to the information we already know, right? And so I think that's totally fine. I mean, again, this is preliminary work. Maggie's not ready to publish on this, but I do think, given the amount of time that she spent on this, I think that this is very nice. All right, guys. I have to run to another meeting, so I think we can break here. Again, Maggie is on. Are you on the GenPALS Slack, Maggie?
Yes, I am.
Maggie's on the GenPALS Slack. She's also on the MCSB Slack and stuff like that. So you can reach out to her. And yeah, thank you for coming. Our next talk is in a couple of weeks, 2 weeks, and it's going to be on spatial transcriptomics.
Cool.
Very exciting. We have one of our amazing spatial transcriptomics people giving a talk, so you don't want to miss that. Okay, thank you, everyone. Have a good night.
Thank you.
All right. Thanks, everyone.